IB Bio, Cell Structure and Function Part 7, will have its focus on the movement of materials across membranes. The essential idea is that membranes control the composition of cells by active and passive transport. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused here. This is the second of two movies on the cell membrane. This movie focuses on mechanisms by which materials are transported across membranes. The fundamental structure of the fluid mosaic model, the Singer-Nicolson model of the plasma membrane, is a phospholipid bilayer. The amphipathic nature of phospholipids allows them to form in bilayers whose relationship to water on either side provides stability, provides integrity. Here's an electron micrograph of a membrane. Remember, it's polar phosphate heads on either side with a lighter area in the middle because of the non-polar fatty acid tails. The membrane is approximately 10 nanometers in width. This diagram suggests 5 nanometers. Here are the IB syllabus statements that speak to the fundamental structure of cell membranes, describe the amphipathic structure of phospholipids and explain their arrangement in membranes, explain how the hydrophobic and hydrophilic properties of phospholipids help to maintain the structure of membranes. Again, the relationship of the phospholipids to each other within the bilayer and the relationship of the phosphate heads to water on either side provides integrity, provides stability for the membrane. That phospholipids are amphipathic allows them to form bilayers. So here is the IB syllabus statement that provides the primary focus for this movie. Describe and distinguish the following terms: osmosis, diffusion, facilitated diffusion, and active transport. Be precise in your use of words, refer to protein carriers, and concentration gradients, and cellular energy requirements. Remember the Singer-Nicolson fluid mosaic structure of the membrane. The fluid mosaic structure includes proteins embedded in the membrane as well as cholesterol, which we can't see here. The proteins have important roles in the movement of materials in or out of a cell. Proteins are involved in both passive transport, which does not require the expensive cell energy, and active transport, which does require cell energy. In this very nice diagram of a membrane protein, you can see the channel through the protein if you look closely. Proteins allow substances to pass without the expenditure of ATP cell energy. This is called facilitated diffusion, and it's passive. No cell energy is required. As well, proteins could undergo conformational shape change under the influence of cell energy, ATP, to actively move substances across the membrane. This is active transport, and active transport requires ATP. But we've gotten ahead of ourselves. Let's take a step back. Let's define diffusion and examine the circumstances under which diffusion occurs. Diffusion is the net movement of particles from regions of high concentration, here, to regions of low concentration, here, and it's due to the kinetic energy of molecules in random motion. Diffusion occurs with no cell energy, and we frequently refer to the diffusion of particles across cell membranes. Remember, diffusion is passive, no cell energy required. Diffusion can happen directly through the membrane or through a protein. In either case, there is a net movement of molecules from regions of high concentration to regions of low concentration. Particles that diffuse through the membrane directly would be small, mostly nonpolar molecules. Size, polarity, and charge are important characteristics that influence whether a molecule can pass directly across the membrane or require a protein. Large or charged particles require a protein. Here is a nice diagram displaying facilitated diffusion that requires a protein. It is passive, however, no cell energy. You can see the higher concentration of molecules here with a net movement of those molecules from high concentration to low through the protein. Again, facilitated diffusion. The protein channels are specific to the molecule passing. The specificity depends on the charges, on the size, shape, polarity on the molecule. The specificity is much like the specificity of an enzyme and substrate. Nerve cell membranes have proteins that change shape under the influence of charges around them. As such, they can open or close, which is a conformational shape change in the protein, and they do that under the influence of the charge, these molecules, in their surroundings. This is facilitated diffusion. Facilitated diffusion of sodium, as can be seen here, occurs through these voltage-regulated protein channels. Once again, facilitated diffusion requires a concentration gradient, high concentration here, low concentration here, and a protein that provides the channel by which the molecules will pass. No cell energy is required. Facilitated diffusion can be observed in graphical form, as seen here, in a graph that plots rate of diffusion over concentration of the molecule that is diffusing. As the concentration increases, the rate of diffusion also increases. But once the concentration reaches a certain level, the protein channel cannot accommodate more of the molecule. Molecules are moving through the protein as fast as the protein will allow. The term used here is saturated. The proteins are saturated. Thus, the rate of diffusion reaches a maximum value and does not increase higher even if the concentration of the diffusing substance is increased. Now let's look at active transport. Active transport also involves a protein, but in this case, cell energy in the form of ATP is required. The protein undergoes conformational shape change under the influence of ATP to pump the particles across the membrane. With active transport, the proteins move the molecules across the membranes from regions of low concentration toward regions of high concentration. In other words, against the concentration gradient. In other words, in this case, no concentration gradient is required. The active transport pumps molecules against their natural diffusion gradient. Here's the relevant IB syllabus statement. Explain the role of the sodium-potassium ion exchange pump as an example of a protein pump and ATP in active transport across membranes. Here's the protein embedded in a membrane of animal cells called the sodium-potassium pump. This diagram is a time sequence and we'll start here. The protein allows 3 sodium ions to enter binding sites on the protein. ATP is spent to change the shape of the protein. The altered shape changes the affinity for sodium, so the 3 sodium ions exit the protein to the outside of the cell. The new conformation of the protein allows 2 potassium ions to bind to the protein. The arrival of the potassium results in the loss of the phosphate group from the ATP that's been attached to the protein all along, thus altering the shape of the protein, and the 2 potassium ions are deposited to the inside of the cell. The sodium-potassium pump uses ATP to pump 3 sodium ions out for every 2 potassium ions in. Here's another example of hydrogen ion pumping under the influence of ATP. The expenditure of ATP changes the affinity for hydrogen. Hydrogen ion binds to the membrane protein. In turn, this transmembrane, integral membrane protein will undergo a conformational shape change. You can see that shape change here relative to the last slide, and the conformational shape change pumps the hydrogen ion out of the cell. Here is a table that compares simple diffusion to facilitated diffusion to active transport with respect to membrane component, whether there's a protein required or not, energy source for the movement, whether ATP is required or not, and the direction of transport from high concentration to low or not. I'll let you study this table on your own. In terms of the movement of materials in or out of a cell, we've looked at diffusion, we've looked at facilitated diffusion, and we've looked at active transport. The last mechanisms by which materials enter or exit a cell are endo and exocytosis. You should already have a feel for these processes. Here's the important IB syllabus statement for endo and exocytosis. Describe exocytosis as a mechanism to rid the cell of waste or as a mechanism to secrete proteins. Describe the way in which vesicles are used to transport materials within a cell and to the cell plasma membrane. Mention how the fluidity of the membrane allows it to change shape, break, and reform during endocytosis and exocytosis. Here's a diagram of exocytosis that you've seen before. The Golgi vesicle arrives to the plasma membrane, and due to the fluidity of the membranes, they fuse and allow the secretion of molecules to the outside of the cell. Examples of these molecules would be enzymes or hormones. You should remember cells that are active in the secretion of protein into a duct. Cells of the pancreas, positioned around a duct, secrete digestive enzymes into the duct through exocytosis. All of the processes to be seen in this diagram should be familiar to you. This is an electron micrograph showing exocytosis. The fluidity of phospholipid bilayer membranes allows the vesicle to fuse with the plasma membrane, releasing the protein outside the cell. Here are pancreas cells surrounding a duct. Each cell adjoins the duct so that each cell can synthesize digestive enzyme to be released into the duct. This structure of cells surrounding a duct is known as an acinus. The acinus displays beautiful form-function relationship in that each cell adjoins the duct so that each cell can secrete digestive enzymes into the duct. These are Golgi vesicles ready to secrete those enzymes into the duct. Endocytosis also relies on membrane fluidity. The cell membrane pinches in, carrying molecules from the exterior to the interior of the cell. Then the vacuole, say a food vacuole, which is pinched off from the membrane, will join a lysosome, and the enzymes of the lysosome will digest these larger molecules within this vacuole. Depending on the nature of the material being taken in, two terms are used. Phagocytosis refers to the uptake of large food particles, large molecules, while pinocytosis refers to the uptake of particles that are more in liquid form. Here is a diagram of pinocytosis on the left and an actual micrograph on the right where you can see pinocytosis happening here and here and here. Pinocytosis is an important mechanism for moving materials across membranes in the lungs, the kidneys, the liver, and elsewhere. Here's another nice diagram of pinocytosis. Again, you can see the particles entering as the cell membrane pinches in and a vacuole is formed. Pinocytosis is an important mechanism for moving materials across membranes in the lungs, kidneys, liver, and elsewhere. Here is a photograph of an amoeba readying to engulf another cell through phagocytosis. Here is a micrograph of phagocytosis. The cell on the left is engulfing the cell on the right. Soon a lysosome will digest this cell that's been engulfed. Here is a nice diagram of phagocytosis with the large food particle being taken in as the membrane pinches in to form a food vacuole. Keep in mind how the fluidity of the membrane facilitates the pinching off of the food vacuole. In this lower right, we can see 4 time periods during which phagocytosis results in a food vacuole. Here is a photograph of a cell that has engulfed a number of other cells through phagocytosis. These cells will be digested with a lysosome. Hopefully you now fully grasp the processes stated here that are involved in the transport of materials across a membrane. We haven't spoken much about osmosis, but that's coming in the next movie. And that brings IB Bio, Cell Structure and Function, Part 7 to an end.